and there's kind of crosstalk between the secretin and the secreted proteins in that way. So I've already mentioned, getting ahead of myself, mentioned cholera toxin, same with LT. You have this assembly in the periplasm targeting to the secretion system, possibly through interactions with secretin. Now, one thing we haven't mentioned up till now, though, is that in many of these type 2 secretion systems, you have something called pseudopilin. So you have a protein which is homologous to pilus subunits, and it forms a pilus-like structure in the periplasm, but it doesn't go further than that. It doesn't actually go out of the cell. And it's thought that this pilus-like structure forming in the periplasm is involved in actually pushing things through, helping push things through the outer membrane complex through the secreting pore. And rather colourfully, it's been described as a piston for pushing the proteins through. Now, related to the pseudopilin, there is actually a pilus, a real pilus, known as the type 4 pilus, associated with this kind of secretion system. This is found in a wide range of different gram-negative bacteria, and often is a key virulence factor. Meningococci, gonococci, these pili are actually involved in adhesion to mucosal surfaces and play important roles in pathogenesis. The interesting thing about these pili is they have the ability to retract, so pull back, and as they're stuck to a surface, in pulling and releasing and pulling and releasing, they can actually mediate a kind of motility known as twitching motility. Quite distinct from the kind of motility we're going to talk about in the next lecture, which is flagella-associated motility. This is a different, quite specialised form of motility. The other thing that these pili can do is they can actually mediate aggregation between bacteria. So we have the so-called bundle-forming pilus in EPEC. We mentioned that earlier. There's this toxin co-regulated pilus of Vibrio cholerae that actually allow bacteria to aggregate into microcolonies. So instead of just binding to host cells, they're actually allowing bacteria to bind to themselves. So you end up with this big colony hanging on there in one place. So we have this pilus being formed in this example here is drawn from Neisseria. You have this type 2 secretion complex, similar kind of players. You've got ATPase involved here. You've got this producing these pilus subunits, which then assemble into in the periplasm. But then they go through this hollow secretin pore in the outer membrane, and actually then extend outwards from the cell. And there's an adhesion at the end, which is known as pil C. So similar to the pili we already mentioned before, they're being assembled at the down here at the basal end of the pilus, and then the pilus is just getting pushed out progressively as the thing elongates. So the subunits that go into the pilus are secreted through the sec-dependent system, and as I mentioned, they're added to the base of the growing pilus. And there's an interaction here where it's kind of based on charge. So the N-terminus of the last pilus that goes in is interacting with a glutamic acid residue on the new subunit. So there's this charge interaction. Platform protein is pil G, and then there's this ATPase homologous to the other ATPases in type 2 secretion involved in hydrolysis, forcing the pilus out of the membrane, and in comes the next subunit, and so on it goes. Now, as I mentioned, there's this strange phenomenon known as twitching motility, which we see in organisms like Neisseria gonorrhoeae. So the pilus is extended, the assembly ATPase playing a role in that. It attaches then to the host epithelium, to specific receptors, so that the adhesion at the end of the filament is actually targeted to bind to something on the host epithelium. And then there is this retraction ATPase, which pulls the whole pilus back. So that allows movement to occur. So you've got this attachment pulled back, and then the cell is moving along. Quite what role this twitching motility plays in pathogenesis is not clear. I mean, we know it happens, and we know that these systems are important for pathogenesis, but quite how the whole thing fits together, it's not clear currently. But there is obviously a role for these systems in adhesion, formation of microcolonies, and it's possible that there is some kind of sensing going on, that these are actually sensing, when they're touching the surface, pulling back, they're actually generating force, which then may be transduced back into the cell and give signals. And we do certainly, we see that with flagella systems, and it's probably likely to be happening here as well. Another important pilus that's actually part of this family is the so-called TCP pilus, or TCR pilus, or toxin co-regulated pilus of Vibrio cholerae. And this is required for colonization of the intestinal tract. It's important in persistence. It's a bit like a bundle forming pilus. It causes these bacteria to aggregate, and it's also involved in the secretion of this colonization factor, which is secreted by this same apparatus. So this, there's a variety of reviews which I'll put up onto WebCT. This is a figure from one of them, which we'll put up there for you to look at. But this just draws together the similarities between these different systems. So you can see that the type 4 pilus assembly system is effectively a subgroup of type 2 secretion, where you have the secreting pilotins, these BIMPs here, platform, the assembly ATPase. But in addition, you have some additional players here, the retraction ATPases, and you have this pilus being formed there as well, that goes outside of the cell rather than just stuck in the periplasm. Yeah, okay. Well, I've covered most of those points. Okay, so the last system I want to talk about today is the type 1 secretion.